The flu. While it is always a serious seasonal disease, in the past, different strains of the virus turned into deadly killers. But with new advances in vaccine manufacturing and delivery, biotechnology researchers are working hard to help prevent a pandemic from sweeping the globe again. Hi, I'm Dr. David Targan, and welcome to BioBytes. Because the influenza virus is continually mutating, the world sees different strains of the virus every year. The deadliest flu outbreak in recorded history occurred in 1918, when the Spanish flu infected 30% of individuals around the world. By the time the pandemic had run its course, it is reported to have claimed the lives of about 40 million people. So what makes the flu serious one year, but deadly another? One factor is the virus's lineage. Usually, the flu spreads from one species to another before it reaches humans. For instance, a bird flu can transfer to a pig, where it would then mutate and finally infect humans. This transferring and mutating of the flu virus actually gives the human immune system a better chance at staving off the infection, since our bodies aren't well equipped to fight the proteins in the strain of flu found in birds. That's why it caused quite a public health panic when bird flu was transmitted directly from birds to humans in 1997. In fact, an in-depth study of the 1918 flu strain revealed that it strongly resembled avian flu strains. Another challenge in flu prevention is matching supply with demand. Currently, flu vaccines are grown in fertilized chicken eggs, and it typically takes 6 to 9 months to manufacture a batch. Health officials across the globe work to identify the most appropriate strains to include in each year's vaccine, but sometimes there isn't a perfect match. In these cases, the vaccine provides protection, but not always at an optimal level. Moreover, this manufacturing timeline would be unacceptable during a pandemic when massive quantities of vaccine would be needed almost immediately. To address these challenges, scientists are using biotechnology to develop a bacteria-based method for producing flu vaccine. Using recombinant DNA technology, the combining of DNA segments from two different sources, scientists can insert genes from the flu virus into a bacterial cell, which then produces key parts of the flu vaccine. This method will help us produce high yields of flu vaccine in a short time frame. Another way developments in biotechnology are helping us to prevent pandemic flu and combat vaccine shortages is with the use of adjuvant systems in vaccines. An adjuvant system is a compound mixed with a vaccine that helps amplify the immune system's reaction to a disease. Because of the way adjuvant system vaccines stimulate the immune system, they can often help the body fight off many different strains of the same virus. This is particularly helpful when fighting a virus like the flu that is constantly mutating. In addition, adjuvant system vaccines usually require less active ingredient in each dose than traditional vaccines. This means that the amount of vaccine produced will help more people in a pandemic situation. Scientists also are researching antibodies from past flu patients to determine which antibodies effectively combat the flu. They've identified certain antibodies that protected against both the Spanish flu and bird flu in clinical animal studies. With these findings and through developments in biotechnology, we hope to create a universal flu vaccine that will provide long-term immunity against the deadliest strains of flu. Continued work with bacteria-based vaccine manufacturing, adjuvant systems, and antibodies provide us with the hope that in the future, we may be able to prevent the next pandemic before it occurs. For BioBytes, I'm Dr. David Targan.